Okay, the experiment that we are going to do today is our third experiment, and it is on plasmid isolation. And this, for this experiment, what we need is a culture that we prepare a day ahead, and that bacterial culture contains the plasmid DNA in it, and we inoculate Laria broth having antibiotic in that medium so that the plasmid can replicate at high copy numbers. Okay. So what we have got in our hands is the plasmid culture, and we will start proceeding with that for the isolation of plasmid, and the method, it generally requires 3 steps. And 3 steps are dependent on 3 different types of solutions. So first I will describe the kind of solutions that will be using, and then I will describe the function of these solutions to make you clear that how they work to isolate the plasmid DNA. Okay, the first solution in this case is it's a buffer, and it is known as cell suspension buffer. So the basic purpose is to harvest the cells from the liquid broth, which is Laria broth most of the times. We spin that culture, obtain the pellet, and we suspend that pellet into this cell suspension buffer. The purpose is to remove most of the salts and the metabolites which have been released into the medium, okay, and to separate the cells for further processing that requires cell lysis and then the precipitation of the plasmid DNA. So first solution is the cell suspension buffer. Solution 2 is cell lysis buffer. Cell lysis buffer or cell lysis solution, whatever you want to say, is okay with that. This solution has got 0.2 normal NaOH, and it has got 1% SDS in it. The SDS helps to dissolve the cell membrane and the cell wall in case of gram-negative bacteria, and NaOH actually helps in the breakdown of the cell wall. The SDS works better in this higher pH, but the actual function of NaOH is, in fact, to denature the double-stranded chromosomal DNA, which is mostly in the linear form. The plasmid, on the other hand, is in supercoiled situation, and the supercoiled molecules are just like the woven threads, and NaOH does not harm the plasmid DNA, but on the other hand, the chromosomal DNA opens up into single-stranded DNA. So this is the basic purpose, to break the cell wall, open up the chromosomal DNA into 2 strands, and then separate this plasmid DNA in the form of supercoiled DNA. Okay. The third solution, which we use in case of plasmid isolation, is a high salt solution, which is 3 molar sodium acetate, and pH of this solution is from 4.5 to 5.0. The high salt solution, when we add it to the lysed bacterial culture, it precipitates the cellular proteins. When the cellular proteins they precipitate, they also get entangled with the chromosomal DNA and co-precipitate the chromosomal DNA along with them. The plasmid, on the other hand, is very, very small molecule as compared to the chromosomal DNA. And it is also supercoiled, so the proteins usually don't affect the precipitation of this along with the SARS. Okay. So as soon as we add solution number 3, the proteins precipitate the chromosomal DNA. The plasmid DNA remains suspended into the solution. And then we perform a spin at high RPM, like 12,000 to 14,000 RPM, which actually removes all the proteins, separates the proteins from a clear solution. And that clear solution contains the plasmid DNA in it that we can later precipitate with the help of ethanol or isopropanol. Okay. So this is the basic mechanism of plasmid isolation. If you have any question. Okay. So what we will do is that in order to proceed, if you have noted this down on the page, then I will mention the procedure like we will take in an append-off tube, we will take about 1.5 ml of bacterial culture. And then we will spin it. Spin at say 3,000 to 4,000 RPM for 2 to 3 minutes. This spinning will yield the bacterial pellet from 1.5 ml. After we have spun, we will get the pellet in the bottom and we can decant this liquid and again we can transfer 1.5 ml of culture just like this one into the tube to collect the pellet from 3 ml cells. So this is a standard procedure of separating the bacterial cells from the Loria broth. We spin it twice. We take 1.5 ml twice in the tube and collect the pellet from there. This is the pellet which is washed from the salts or any other metabolites, and this pellet is now ready for suspension in solution number 1. After suspending this pellet in solution number 1, which is actually a buffering agent, so we add to this tube, which has a pellet about 200 microliter of solution 1. Okay, suspend it to get a fine suspension of the cells, and then we add about 200 microliter of solution 2, which is having NaOH and STS. And when you add this solution, just gently mix. Mix gently, very gently, okay, and you will see actually that this suspension, which is opaque, it turns transparent. That is an indication that the bacterial cells have been lysed. Once you observe this, and the time is very important here. The time of incubation should not increase. It should not go beyond 2 minutes. And as soon as you see the lysis has occurred, we add solution 3. About 250. Yes. The reason is that if we incubate for more than 2 minutes, the plasmid, which is supercoiled form, it gets more access to the NaOH, and NaOH can damage it, it can open it and denature the plasmid as well. So we want to protect the plasmid from denaturation by NaOH, so we limit the time. That is the reason it is very important to keep that 2 minutes time, not more than that. So after addition of solution number 3, what you will see is what you will see in the tube is that after all this tube is filled with all the 3 solutions, the pellet has been suspended, you will see the precipitating proteins, a whitish specks of proteins which will appear, and that will be the indication. And this protein is very much fluffy. Okay. Now, what you need to do is that after addition of this solution 3 here, spin. And you need to spin it at high speed, the maximum speed which is allowable by your microfuge. That is usually 12,000 to 14,000 RPM. And this can be done for about 10 minutes. I said that this protein precipitate is very much fluffy, and it is very hard for this protein to get it precipitated. So it will not form a pellet, a true pellet. But what you can see is that after spinning, you can see the protein is somewhere here, the supernatant, the clear supernatant is here, and some turbid materials is at the bottom of the tube. So you need to take this clear supernatant into a fresh tube and measure the volume. And the volume would be around 400 to 500 microliter that you can collect from here, the supernatant. The next and the final step is the precipitation of plasmid DNA. And the plasmid DNA from the supernatant that you collect from the top of the tube after spinning it can be obtained by adding either ethanol or isopropanol. Precipitation of plasmid it can be achieved either by adding ethanol or isopropanol. Ethanol is added at 2.5 volumes and isopropanol is added at 1 volume. It means that if you have collected 400 microliter of the supernatant from after the salt precipitation, okay, you have 400 microliter of the solution here, you can add to it 800 microliter to 1 ml of ethanol. or alternatively, you can add to this tube, which is having 400 microliter solution, supernatant, you can add 400 microliter of isopropanol. Once you have added these, either you have used ethanol or isopropanol, mix that solution thoroughly, and you can keep these tubes at -20 degrees centigrade for about 30 minutes. This incubation at -30 will actually precipitate the plasmid, and you can see turbidity inside the tubes, which is an indication that the plasmid is being precipitated. So finally, for the isolation of plasmid, once you are done with all this, you can spin that tube in a microfuge. (10,000 to 12,000 RPM). And what you get is, this is the turbid solution having the plasmid DNA in it. And after spinning about 10 minutes, you will find that there is a very fine white pellet at the bottom of the tube and decant all the liquid from the top. You can wash this pellet with cold ethanol. Okay, that's usually 70% ethanol. You can use cold ethanol to wash this pellet once or twice, and then dry the pellet by inverting the tube on a tissue paper, draining all the liquid, and keeping the tube for a couple of minutes on your bench top. Okay, and you can blow a little bit to evaporate the ethanol. Okay, and depending on the size of the pellet, you need to decide how much volume of water you should use to dissolve this pellet. Okay, so after drying and cleaning of this pellet, you need to select from 10 to 20 microliter of water. So this pellet is usually dissolved in 10 to 20 microliter of water, depending on the size. And this size is dependent on the state of the culture that you obtained actually in the beginning. If your growth of the bacterial cell was confluent, like 0.7 to 0.8 optical density, so you will get enough plasmid DNA for suspension in 20 microliter of water. But if your optical density is lower than that, then you have to use a lower volume so that the concentration of the plasmid should be high in a lower volume. Okay, so this is how we are going to proceed on the bench, and now we will see that how we do all these steps to recover that plasmid, and I will show you each of the steps one by one as discussed here on the board. Thank you so much. And let's go for plasmid isolation. Okay, we are now in the laboratory and we are ready for taking the experiment of plasmid isolation. I will introduce you to different equipments that we need for plasmid isolation. And here we have got a microcentrifuge machine, which can go till about 14,000 RPM. And we can adjust that speed through this knob. And we can see the speed dial, which is here when it is running. It is set presently at 12,000 X, but we will be using it maybe at higher speed later on. Next we need is a pen of tube rack, where we will keep our pen of tubes and take our liquids, especially the bacterial culture, and then the addition of solution 1, 2, and 3 will be here. These are some of the culture tubes which are carrying the solution 1, 2, and 3. So we will just pipette out some of the liquids from these tubes. We need the append of pipettemen set, and we have here 3 pipettemen which are 1 ml, 200 microliter, and 10 microliter each for various kinds of volumes that we will adjust during this pipetting for the plasmid isolation. And these pipettemen will utilize the micropipette tips. These white ones, they are the smallest one, and they can measure the volume like 10 microliter or 20 microliter very easily. The next one is the yellow tips, which are usually 200 microliter, but we take generally 100 microliter of the volume through these pipette tips. And then now you see the 1 ml tips, which are usually known as blue tips, okay? And we can take up the volume up to 1 ml in these tips. So we have our cultures ready here, and what you can see on the left hand side is actually the Loria broth medium, which does not have any bacterial growth in it. So this is the Loria broth which we inoculated yesterday with the bacterial cells having plasmid in them. And if you see now on the right hand side, these tubes, they are very well turbid and carry the bacterial cells which we are going to harvest for plasmid isolation. So you can clearly see the difference in the tubes having growth and no growth. And we will be utilizing these microfuge tubes for taking the materials in them. First we will take the bacterial culture, and we will follow the protocol as was discussed in the introduction section. And this is the first part of the experiment where we will actually isolate the plasmid DNA, and then we will go for seeing the plasmid DNA through electrophoresis. And here we have got a power supply and a horizontal gel electrophoresis apparatus. So we will discuss everything one by one, and let's now start with the first step of plasmid isolation. Okay. So this is the beginning of the experiment on plasmid isolation. Aruj is taking the liquid culture out of the tube into the append of tubes, and just I will show you that how she is doing it. She has taken a 1 ml pipetteman, and she is pipetting out the liquid from the culture into the append of tube. So this is about 1.5 ml that she has taken in the append of tube, which is already graduated, and you can actually follow. Can you show me a close look of that? Yes. So you can see that this tube is actually carrying a 1.5 ml of the liquid culture and now what we are going to do is that we are going to spin tubes after proper balancing in that machine. So she will take some liquid in the second tube, that will also be 1.5 ml. Okay, she will use this tube actually to balance the first tube that she has already placed in the microfuge, and then we are going to spin this.
Yes, so one of the tube is a balanced one and the other carries the bacterial cell culture. They are placed opposite to each other. After keeping the lid, she's adjusting now the RPM on the machine using this knob.
4,000?
Yes, you can keep to 4,000 is enough for that. For the bacterial cell pellet, 3,000 to 4,000 RPM for 2 or 3 minutes is fine. Yeah, you can now start spinning. So it's running at 4,000 RPM and the time is there for 3 minutes. So we will come back to the machine in 3 minutes and we'll take up the tubes, decant the supernatant, and we will put another 1.5 ml in the same tubes. Yes, the 3-minute time is now over and the machine is stopping down. It's coming down back to 0 after these 36 seconds. So we will have a look at the pallet and see how big it is. Aruj, can you bring some tissue paper to drain the tube completely? Yes, the machine is now stopped and she will wait to open this. Can you see the pallet here? Okay, yeah, I can see a little pallet down there. Yeah, you can see actually the pallet. It is right there. So this is a small pallet. We will spin 1.5 ml more into the same tube and we will get a bigger pallet to get a good isolation of the plasmid DNA. That's perfect, Aruj. You can proceed with, you can drain this now. So you see how she is draining complete liquid. Yeah, it's perfect. Yeah, proceed further. Another 1.5 ml from the bacterial culture. Another spin at 3 to 4,000 RPM for 2 minutes or 3 minutes will be great. And you will see that the pallet size will increase significantly and will assure a good isolation of the plasmid DNA from there. So it's running at 4,000 RPM for 3 minutes. So we'll wait for the centrifuge to stop and take out the cells from there. So this machine is going to stop now, and when the RPM is 0, you can hear the click. You can open the lid now and you can take out the tubes. Now I would like to see the pallet again, Aruj. If you can bring it a little, yeah, it's perfect. I can see the pallet. It's quite bigger than before, and I hope you can see it very well. I can see that. Yeah, you can drain the liquid from there. And we will begin with solution number 1. That is the cell suspension buffer. She's just draining the lauria broth and the metabolites which are there in the lauria broth so that we can have a pure suspension. Yeah, we can see the pellet actually from far distance. Yeah, perfect. So this is solution 1. Can I see the label? Yeah, this is solution 1, which is the buffer. So she will take about 200 microliter from this solution and it will be dropped over the pellet. And you can now resuspend the bacterial cells by tapping or by pipetting in and out of the solution. Can you hold it in a way that I can picture it? Yeah, it is fine. She is pipetting in and out, and you can see that the cells are being suspended and you are getting a turbid solution. Yeah, 200 microliter of the cells. That's perfect. So after you suspend this bacterial pellet, you can see the turbidity actually in the cells, which is whitish now instead of a yellowish solution. Okay, so now you can add solution number 2, which is actually the lysis solution, to this one. Okay, now you can see actually the bacteria suspended in the buffer. And now she will add some lysis buffer in it. Yes. And I will check how we can monitor that it's getting lysed and gets transparent. Can you add the solution 2, please? Just do it slowly so that I can keep it seeing that how the transparency comes up. Yes, right away we can see that the solution is getting clear, and it will be more clear as we mix it, but mix it very gently. Can I see it? Can I see that? Yeah, it's now lysed. Can I see that too? Yes, it's lysed. It's lysed. Can you open the cap? I want to see if there is a sign of lysis in there. Okay, the suspension is much clearer as before. Can you suspend it a little bit more? I want to see whether it gets a little bit more clear or not. Yeah, just gently, very gently. Yeah, it's perfect. So I think it's already more than 1 minute. Okay. Yeah, it is quite clear than before. Now you can add actually solution number 3 to it. And this solution 3, I hope you remember, is 3 molar sodium acetate, which is a very high concentration of salt. So as soon as you put this salt, can I see the tube now after putting the salt? Aruj, can you bring it a bit closer? Yeah, it's fine. Now you can see how whitish precipitate is being developed at the meniscus of solution 3 and the plasmid solution which we obtained after cell lysis. Can you mix it just gently, Aruj? very gently, yeah. It's getting whitish. And let's see if we can see the precipitates in a while. The proteins are getting precipitated and the chromosomal DNA will be trapped in the, yeah, it is very much white now, which is a very clear indication, in fact, that the proteins are getting precipitated. Yeah. Yeah, but it's quite whitish now, which indicates proteins are there. You can mix it a little bit more. Okay, that's perfect. And now we can go for spinning this tube at the maximum speed so that we can get rid of this whitish precipitate and get the clear supernatant for precipitation of. Okay, so you're balancing the tube?
Yes, sir.
Okay, and after balancing, we will put it at spin, you know. We will see what is the maximum RPM of this machine. So if we can go at a maximum RPM, it will give us a great separation of the proteins from the solution. So stay at 13,000 for 10 minutes. Okay, so turning the speed, so you can hear the noise. The machine is running at full speed, that is 13.3 thousand RPM for 10 minutes, and we will come back in 10 minutes to this machine to take out our samples. Okay, the machine has stopped now and we can take out the tubes. This is the situation that the protein has been precipitated and it is in the bottom. So we can take up the supernatant, which is quite clear, from this tube into a new tube. So it's going to be 400 or 500 microliter of the supernatant. And then she will add an equal volume.
400 microliters.
Okay, so it's 400 microliter. So we can add about 800 microliters to 1000 microliter of ethanol into it. That is 2 to 2.5 volumes of ethanol. Ethanol evaporates very well, so we usually prefer ethanol for the precipitation of DNA, whether it is plasmid or chromosomal. Yeah, can you mix it gently? I want to see that if we can see some swirls or whitish. Yeah, there's a slightly whitish turbidity in this mixture now, which is an indicative of the fact that the plasmid DNA is being precipitated. It's either very small amount, but we can see there is a whitish tinge coming up in the tube after the addition of this ethanol in it. Okay, so after mixing it, we can keep this tube at minus 20 degrees centigrade for about 30 minutes. Okay, and after 30 minutes we will give it a spin again to precipitate the plasmid DNA. Okay, go ahead, Aruj, keep it at. We have taken out the tubes from freezer. We placed them at minus 20 degrees centigrade. There were 2 samples, okay? And now we are going to give these tubes a spin to pellet the plasmid. So you can proceed further, Aruj. Yeah, you can give it a spin at full RPM for about 10 minutes. That is 13,300 RPM. That's the maximum speed of this machine. So we'll come back in 10 minutes. The machine has stopped, and now you can take out actually the tubes and let's see what kind of pellet it has got. So, can you see that? Can you tilt? Yeah, it's at the bottom. I can see that. The whitest speck that can be seen at the bottom of the tube. Arush, can you point out onto that pellet that is? Yeah, that's visible. That's quite visible, and usually this is the size of the plasmid pellet that we get from 3 ml of the cell culture, and this is a typical size of that pellet, and we can suspend this pellet in 15 to 20 microliter of water. Later on, after washing it, can you decant the solution from here? And this is now the ethanol washing starts. We can use 200 to 300 microliter of ethanol for washing this pellet, just to remove the salts that has been incorporated from solution number 3. And so what she has done is that she has added ethanol and mixed it a little bit and going back to spin, because this ethanol makes this pellet a little bit loose. Okay, so just to make a firm pellet again, she will give a quick spin of this at full RPM. This is just a quick spin. You can stop it now, it's enough. The pellet will get settled at the bottom of the tube, and we can decant the ethanol. I don't think we need to rewash it with ethanol. Just we will drain and evaporate the ethanol, and we will suspend this pellet in about 15 to 20 microliter of sterile double distilled water that we have already purified, and it is kept at minus 80 degree centigrade or minus 20 degrees centigrade. And we take the tube out just prior to use. So this is taking out the ethanol from the tube. So you can decant if there is any remaining material onto the tissue paper by inverting the tube on that. On the tissue paper, yeah, you can see the pellet very clearly, visible. Yeah, you can see the plasmid DNA. Yep, and it does not have any liquid. She has removed it completely. We will dry it for 1 or 2 minutes, or you can give a slight blow. Slight blow into the tube just to evaporate the ethanol, okay? And we can notice that the presence of ethanol by smelling this tube. So if it still smells for ethanol, we can blow it a little bit more, okay? And then the pellet is ready for dissolving in the double distilled water that I was mentioning. It is sterile and it's kept at minus 20 degrees centigrade. We take a tube out and we take just 20 microliter into this tube for dissolving. You can dissolve the pellet now. And our plasmid is now ready for loading onto the gel after we have dissolved it in water. After dissolving this plasmid, we need to actually keep that in ice, or we can keep that in the freezer at minus 20 degrees centigrade. Okay, the pellet immediately dissolves up. It pops up in the water and it starts dissolving itself, giving us a clear supernatant without anything in it. Yep, it is done.